Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, today we are going to begin with very important topic, biomolecules. Okay, biomolecules, they are termed as these chemical substances, molecules. They are found in biological system. So these molecules, whether they are of any category, they are very essential for purposing the life, for carrying out different metabolic reactions. So starting with the first slide, what are the objectives of this chapter? At the end of this chapter, you will be able to distinguish between several molecules like micro and macromolecules. Okay, primary and secondary metabolites. The chapter will help to realize the importance of these molecules, okay, in the cellular, intracellular, as well as extracellular life. Learn all the compounds that are present in the cell. The science which deals with the study of chemical constituents and the reactions between them in any living organism is called biochemistry. So biochemistry, it is a science which deals with the study of chemical constituents and the reactions between them in any living organism is called biochemistry. Newberg used the term biochemistry for the first time and is also known as father of biochemistry. Father of biochemistry, Newberg. The cell's protoplasm has organic as well as inorganic matter. Mean the protoplasm of a cell is composed of both organic as well as inorganic matter. Both of them, they are termed as biomolecules. So both organic and the inorganic matter, they are known as what? Biomolecules, right? These biomolecules, they help the life to become possible as they carry out different metabolic reactions. The cellular biomolecules together are also called cellular pool, which includes water, inorganic materials, and organic materials. So the cellular biomolecules, it is also known as the cellular pool. It helps us to create, or it helps the cell to create a system having all the cellular components and the organic, inorganic, water, all these components together, right? There are various types of divisions of biomolecules. Biomolecules are classified on different bases. Now coming to the next slide, the elements and their relative abundance. Hydrogen in the earth crust, its weight is 0.14% only, human body 0.5%. Carbon 0.03%, human body 18.5%. Oxygen 46.6%, most abundant, and in human body 65%. Nitrogen traces, human body 3.3%. Sulfur 0.03%, human body 0.3%. Sodium 2.8%, in human body 0.2%, but very essential. Calcium 3.6, 1.5%. Also, it is also very essential. Magnesium 2.1, 0.1. Silicon 27.7, second most abundant element after oxygen, okay, and negligible. So now we are going to discuss about micromolecules and macromolecules. Micromolecules and macromolecules, they are basically the classification of biomolecules depending upon their weight, concentration, their structure and solubility. If the concentration, weight, structure and the solubilities of the molecules in a cell differ for all the types, okay, the molecules which have low molecular weight, simple structure and higher solubility, they are known as what? Micromolecules. Okay, so micromolecules and macromolecules, it is basically a classification which is categorized on the basis of concentration, weight, structure and solubility. If the molecules which have the low molecular weight, simple structure and more solubility are known as micromolecules. Example: minerals, water, sugars, especially the simple and the complex sugars, and amino acids. The chemicals that have higher molecular weight, lower solubilities and complex structures, these are the polymers, biopolymers or biopolymers. These are known as what? Micromolecules, like proteins, nucleic acids. Okay, so they are so complex that their solubility is less in comparison to the micromolecules, right? Now coming to next, organic and organic compounds. Organic compounds, they are derived from organisms containing carbon, hydrogen, and along with carbon, hydrogen, nitrogen, sulfur, oxygen, phosphorus can be there. Inorganic compounds which are formed in the form of minerals, and they are most abundantly found in earth crust, right? The molecules that have carbon, hydrogen, oxygen together in their composition is called organic compound, like carbohydrates, proteins, fats, nucleic acids, enzymes, hormones, etc. They are organic compounds. The molecules that do not have carbon, hydrogen, oxygen as a group in their composition is called as inorganic compounds, like minerals, water, CO2, N2O, etc. They all are inorganic compounds. The organic compounds, they are basically derived from the organisms. Either they are primary or secondary metabolites depending upon the extraction. And the inorganic compounds, they are the molecules that do not have carbon, hydrogen, oxygen as a group, as a repeating unit. So they are known as what? Inorganic compounds. They are not derived from organisms. Okay. Examples are minerals, water, etc. Okay. Now coming to next, major elements and minor elements. Major elements and minor elements are categorized as the elements that are higher in amount in a cell, in their concentration in cell is more. They are known as what? Major elements, like carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. So they are major elements because they are abundantly found in, the cellular cytoplasm. Now, together, all these major elements make 98% of the composition of protoplasmic weight. Thus, they are known as protoplasmic elements. They are known as what? Protoplasmic elements. Minor elements are the chemicals that are less in amount in the cell: calcium, magnesium, potassium, chlorine, iodine, iron, sodium. They make around 2% of the weight of protoplasm. So the major elements which are found in higher amounts, minor elements which are found in lower amounts. Minor elements mainly includes the inorganic materials like calcium, magnesium, potassium, chlorine, iodine, iron, sodium, etc. The composition is very much variant. The minor elements, they constitute only 2%, while the major elements constitute 98%. Okay. Now, here only I would like to distinguish between the primary and the secondary metabolites, compounds which are present in animal tissue or which are present in animal tissues. They can be directly extracted in their native form. They are known as what? Primary metabolites, right? Primary metabolites. In secondary metabolites, the primary metabolites showing there like generally compounds present in plant tissues. They are secondary metabolites. Okay. So these secondary metabolites includes alkaloids, rubber, essential oils, antibiotics, etc. Okay, these are main compounds which are present in which are seen in animal tissues. They are primary metabolites, and which are seen in the plant tissues. They are secondary metabolites. Okay, secondary metabolites, they have ecological importance. Ecological importance. Now, the cell usually is of micrometer in size, which is enclosed in a membrane or a wall. Thus, to study its constituents, we need to open the cell and expose or reveal its compounds. That's right. The cell is hence extracted from the source, plant stem, animal liver, microbial colony, etc., and then mixed with trichloroacetic acid. This mixture is ground with pestle and mortar, which is further stained through cheese, cloth, or cotton. So the cell is extracted from the source like plant stem, animal liver, microbial colony. Then it is mixed with trichloroacetic acid, which gives a cryogenic pressure. This mixture is ground with pestle and mortar, which is further strained with cheese and cotton. The resultant has two fractions. After getting cold treatment with trichloroacetic acid, the two fractions are the filtrate, which is called the acid soluble pool, while the residue, which is called the acid insoluble fraction. The filtrate, which is known as what? The acid soluble pool, and the residue, which is the acid insoluble fraction. The acid soluble pool, the acid pool includes cytoplasmic composition. The cytoplasm and the organelles have macromolecules, also known as biomacromolecules, which are insoluble in acid and thus are present in the residue. So the resultant has two fractions. One is the acid soluble and the other one is acid insoluble fraction. The acid soluble fraction, it includes the cytoplasmic composition, which includes cytoplasm and the organelles having biomacromolecules or large molecules which are insoluble in acid and thus they are present as residue. The fractions obtained are further separated to identify and to study the components of the cell by various analytical techniques. The conclusion from the method used are that the cell has both small and large molecules present in it. Okay. The filtrate is found to have small molecular weight chemicals called as biomicromolecules and large molecular weight chemicals known as biomacromolecules. Okay. So how will you extract? You have to take out the cell and then you have to mix with the trichloroacetic acid for cryogenic treatment. Okay. It helps to crystallize the the macromolecules. Okay. There are some important facts or points that protoplasm is a Greek word. Protos means first and plasma means organization. Cotai in 1772 first observed protoplasm. Felix Dujardin called it as sarcode in 1835, who observed the animal cells have jelly-like substance, protozoa. J.P. Purkinje in 1840 studied plant cells and found the similar substance. He also coined the term protoplasm. Hugo van Moll in 1846 studied the nature of the embryonic cell protoplasm of plants and also explained the protoplasm importance of cell division. Okay. Max Kuhls in 1861 studied the sarcode, which was given by Felix Dujardin, and protoplasm and led their similarities. Schultz proposed protoplasmic theory, name given by O. Hartwig. Max Schultz called protoplasm as physical basis of life. So Max Schultz, he called protoplasm as physical basis of life. J. Purkinje, 1840, he compared the substance and found or coined the term protoplasm. Cotai in 1772 first observed protoplasm. Okay, but the term protoplasm was given by the J. Purkinje. Okay, Hugo van Moll, he studied the nature of embryonic cell protoplasm and also explained the protoplasmic importance in cell division. Schultz proposed protoplasmic theory, name was given by O. Hartwig. Max Schultz called protoplasm as physical basis of life. J.S. Huxley in 1868 wrote the book and published his book Protoplasm as Physical Basis of Life. Rinke and Roderwold in 1881 first of all performed the chemical analysis of protoplasm. So the major points are the chemical analysis of protoplasm, which was proposed by Roderwold and Rinke. Okay, in 1881, J.S. Huxley in 1868 he published his book Protoplasm is the Physical Basis of Life. Cotai, J. Purkinje. Okay, and Max Schultz. Physical nature of protoplasm. Theories that are proposed which explain the physical nature of protoplasm. Alveolar theory given by Boucher. Granular theory given by or proposed by Altman. Fibrillar theory given by Fleming. Reticular theory given by Henstein, Klein, and Carnoy. So Henstein, Klein, and Carnoy, they all proposed reticular theory. Fleming proposed fibrillar theory. Altman proposed granular theory, and Bush proposed alveolar theory. The above listed theories were not accepted. Colloidal theory, Fishy-Hardy, and Wilson, they proposed colloidal theory. This is the most acceptable theory of protoplasm in a cell. According to this theory, the protoplasm is a polyphasic colloidal system. Sol gel. Compounds of protoplasm. The chemicals in the cytoplasm are present as free ions or as the combined form which two or three different elements form. Okay, so these ions they make the compounds. Okay, so the inorganic compounds in the protoplasm are water 70 to 90%, salt, acids, bases, and gases around 1 to 3%. So water is 70 to 90%, while rest over the salts, acids, bases, gases, okay, they are around 1 to 3%. Organic compounds present in the cytoplasm are proteins 7 to 14%, lipids 1 to 3%, carbohydrates 1 to 2%, nucleic acids, enzymes, and other 1 to 3%. The dry weight of animal cell is around 60 to 70% part of protein, okay, having protein. The dry weight of plant cell having 80% of the carbohydrate, okay, in plant cells, because the cell wall is made up of carbohydrate, cellulose, complex carbohydrate. So the dry weight of animal cell is made up of 60% protein and dry weight of animal cell is made up of 80% carb, as the cellulose is a complex sugar. Understood? Is that understood? Yeah. Now the major composition is water 70 to 90% and then protein 7 to 14%. Carbohydrates 1 to 2%, lipids 1 to 3%, nucleic acids, enzymes and other 1 to 3%. Okay. Now carbohydrates. Carbohydrates, they are the first respiratory substrate for the oxidation. Okay. Carbohydrates which produce maximum energy in the body. So after getting oxidized or after undergoing the cellular respiration, they produce maximum energy. Maximum energy or readily delivered. Very fast. They have carbon, hydrogen, oxygen in a ratio of 2 is to 1 is to 1. Okay. When the ratio of hydrogen and oxygen is 2 is to 1. And thus they are also called as hydrates of carbon. Okay. So remember this ratio only. The ratio of hydrogen is to oxygen in carbon, hydrogen, oxygen is 2 is to 1. And that is why hydrogen is to oxygen ratio 2 is to 1, it resembles or it replicates that they are formed by water. Okay, they have hydrates with water in them. So that's why they are known as what hydrates of carbon. Generalized formula of carbohydrates is CxH2O whole by their X and Y are the real natural numbers from 1 to 3 when all the numbers are quantized. They follow the law of chemical combinations. They are simply carbohydrates and complex carbohydrates. The simple ones are soluble in water and tasty. Okay, and they are sweet in taste with their sweetening index is high, which are called sugar. The complex ones are insoluble in water and they have no taste at all. The simpler ones, they have high sweetening index. The structure of simple carbohydrates has an aldehydic group and the complex carbohydrates has ketonic group. The structure of simple carbohydrates is having an aldehydic group and the structure of complex carbohydrates they have ketonic group. Please remember this form that carbohydrates they are of two types, aldose and ketones. So the complex one they have ketonic sugars or the ketonic carbohydrates and the simple carbohydrates are aldose sugars. Understood? Classification of carbohydrates. The saccharide number based on the saccharide number. Okay, the carbohydrates are classified as monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides general characteristics are that these are simplest sugars which cannot be further hydrolyzed and which cannot be further broken down as they have only one saccharide unit. Saccharide unit is the repetitive unit of sugar, simple sugars. Okay. The formula is CnH2O whole N for the monosaccharides. Monosaccharides occur in D and L absolute conformations, D and L relative conformations, sorry, relative conformations. Okay, with the exception of dihydroxyacetone, which does not have chiral carbon in its structure. Chiral carbon means asymmetric carbon. It is the carbon in which all the four groups are different. Chiral carbon is a central carbon which has all its four valencies satisfied by different functional groups. So monosaccharides they occur in D form and L form, that is relative configurations. Okay, with the exception of dihydroxyacetone, as it is not having any chiral carbon or asymmetric carbon. Okay, that is why it cannot show the relative configurations. Okay, conformations. The chiral carbon is the central carbon which has all its four valencies satisfied by four functional groups. Okay, different functional groups. This is the structure of glucose. You can see the open form, ring form. Okay, so we will be discussing all these forms in the coming lectures. I will discuss all these forms in the coming lectures in detail. Till then, bye bye, take care, stay safe.